Hello everyone and welcome to the NPTEL course on pharmacognosy and phytochemistry. Today I, Galvina Pereira, Assistant Professor, Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai, will take you through a set of very interesting compounds that are found in nature, and those are tannins. Now what are tannins? Tannins derive their name from tanna. Tanna is a German word for oak which was used in tanning industry. Tanning converts the animal hide into leather. This happens because tannins have the ability to precipitate animal hide proteins and convert it into stiff metabolites. These stiff metabolites prevented putrefaction. So tannins are astringent. By their chemical nature, they are polymers of polyphenolic compounds, especially acids, biomolecules, and have a typical astringent effect. If you see tannins, tannins have a huge molecular weight. The molecular weights of tannins range somewhere between 500 to 3000 kilodalton, and in some cases when they form complexes or adducts with other metabolites, their molecular weight may be as high as 20,000 kilodaltons. Tannins in plants are generally born or generally biosynthesized out of a unique pathway called as shikimic acid pathway. This pathway is unique for a reason. It is found in plants and microorganisms, but it is not found in humans. A very simple example of a tannin derivative is a structure you see here called as gallic acid. It is trihydroxybenzoic acid. It is the simplest form and simplest monomer of tannin. This may undergo polymerization to form a high molecular weight polymer of tannins. Now let's see some day-to-day examples. Tannins are compounds which render a unique tan or brown color to compounds. Just to state a few, a tea that you have on your day-to-day basis, your chocolates, deep brown color of your dark chocolate, be it coffee, be it your Clubbird or your nut gulls. All of them have a unique pigment which is deep brown in color, and those are polymerized tannin. When they are not polymerized, they are little lighter in color, and as you can see amla has tannins but in a small molecular weight range. So if you take amla as an example, when you consume amla, you get a typical tactile sensation termed as astringency. So this tactile sensation is often attributed to presence of tannin compounds which are there. These tannins mix with your saliva. They will precipitate the compounds or especially proteins and enzymes which are there in your saliva, and this colloidal precipitate in your mouth will give you a frictional, dry, rough, often referred to as a puckering feeling. Now this is what is called as astringency. So this colloidal tannin precipitates then make your tongue little rough, and then that sensation remains for a while till the tannins are kind of digested by your saliva and then ingested by us. So they form precipitates with salivary proteins, and if you see this, the degree of salivation in individuals vary. So people producing more saliva, they'll have definitely more proteins, they'll get the sensation a little less. But in some cases when your amount of saliva is less, you will get a very rough feeling after consuming astringent substances which contain tannin. Now why do plants synthesize tannins? Tannins have a very important role to play in plants. Now let's take an example of our skin. The moment we walk into the sun, what happens is, to prevent the deterioration of our delicate molecules that are there in our skin, the skin will synthesize protective molecules called as melanin. This melanin serves to protect our skin from the harsh radiations of the sun. Similarly is the role of tannins in plants. Whenever the tree is exhibiting any harsh sunlight, it will produce more of this polyphenolic compounds, and as a result you see, if you want more tannins, especially an example is your tea. Those tea plants are often planted in a very sunny, hilly area where the exposure to sunlight is maximum. The second role that it plays is it prevents infections in plants. Now we know polyphenols such as cresol and other phenolic compounds have a very potent antibacterial or fungal effect. Plant produces similar polyphenols to get rid of bacteria and fungi. Whenever a tree is injured, what is going to happen is it is prone to infestation by bacteria and fungi. So to prevent that, plant will go on synthesizing these polyphenols and it will secrete in the region where it is wounded. And as a result, these proteins of the bacteria and fungi are precipitated by the tannins, thereby killing the bacteria and fungi. But over a period of time, many bacterial and fungal species have started producing an enzyme called as tannase, which cuts off or cleaves the tannins, and they have become resistant to the effect. Not only that, their astringency deters bigger insects or animals from consuming it. So especially in the young stage when the tannins are in a low molecular weight stage, in that case you will see that the animals are deterred because of its astringent effects. So properties of tannins. Now tannins are amorphous in nature. So what happens here is your tannins are large molecular weight compounds. They are polymers, and if when you see a polymeric stage, the degree of crystallinity reduces, and as a result most of the tannins are amorphous in nature. Now because of their large molecular weight and polyhydroxy groups, they are not soluble in organic solvents, but they are rather polar and dissolve readily in water. With exception of very high molecular weight structures, many of the tannins are water soluble, ethanol soluble, soluble in acetone and glycerine. They have a unique ability to combine with proteins. The hydroxy group of tannins combine with the amine group of proteins to form complexes. So same lies true for gelatin, which is an amine containing polymer. It has also ability to complex with cellulose and pectin. Those are the sugar derivatives, and has a unique ability to chelate with metals. Now this chelation often leads to formation of colored complexes. So if you see tannins reacting with metals such as iron, it will give you interestingly blue-green color complex. In addition to that, being polyphenolic in nature, tannins show antioxidant properties. Here is an example of few tannin monomers. Just like we discussed, tannins are actually polymers. But what are the building blocks of tannins? So the building blocks of tannins are monomers, and the simplest monomers we just discussed is a trihydroxy benzoic acid derivative referred to as gallic acid. The second monomer, if you see and if you carefully cut it from here, so if you just cut this molecule from here, you can identify two gallic acid molecules. Okay. So here is the first gallic acid and there is the second gallic acid residue, which are joined to each other by a C-C linkage. Now what happens is, this is what is called as hexahydroxy. There are 6 hydroxy groups, 1, 2, 3, and there is 4, 5, 6 representative of gallic acid. So hexahydroxy diphenic acid. Eventually what happens is, this may undergo a water elimination to form lactone, and that lactone is called as ellagic acid. Now let me just quickly draw it for you. It's a bis compound, so there are 2 phenyl moieties, each representing a phenyl of gallic acid. Now trihydroxy, and this is the acid group. So I'm just keeping it as such. This is the acid group. I'm just keeping it as such, and let's put the hydroxys here. Okay. So what is going to happen in the process is when I draw this carboxylic acid like this, I have two gallic acid residues. They undergo water elimination to form what are called as depth side linkages or lactone esters. So this are form lactone esters, and this is called as ellagic acid. So hexahydroxydiphenic acid undergoes lactonization to form ellagic acid, which is another monomer. The next set of monomers, which are your benzopyrone derivatives, more specifically phenyl benzopyrone derivatives, and the numbering goes like this: 1, 2, 3. So they are called as flavan 3-ols. In some cases, an additional hydroxy is found at the fourth position. In that case, they are called as flavan 3,4-diols. The last set of compound you will find are actually compounds called as phenyl propanoids. So there's a phenyl ring, there's a propane 1, 2, 3. So this is a typically cinnamic acid derivative which forms ester with quinic acid, and you will get what is called as chlorogenic acid. So if you see tannin monomers, they are small molecular weight. They may undergo polymerization to give what are called as true tannin derivatives. So how do you classify tannins? Those with small molecular weight monomers are called as pseudotannins for the reason they cannot bind to the animal hide and cause what is called as the binding, stiffening or tanning of leather. So they do not tan leather, and hence they are called as pseudotannins. On the other side, when they themselves polymerize all these monomers, they give what are called as high molecular weight complexes. They acquire the ability to tan leather, and in that case they are called as true tannins. Now true tannins can be classified into many classes depending upon their susceptibility to hydrolysis. The simplest ones are hydrolyzable. Now hydrolyzable, as the name indicates, the ester linkages in them can be hydrolyzed by acids, by bases or by enzymes we just saw, that is your tannase. Depending upon which monomer is involved, if it is a hexahydroxydiphenic acid or ellagic acid, we call such tannins as ellagitannins. If it is a gallic acid monomer, we call it as a gallotannin. If it is a mixture of both, that is ellagi plus gallic acid together, that is if it is a mixture of both ellagic acid and gallic acid put together, we call it as a combined or both tannins. Now if you see the hydrolysis, there are another category called as non-hydrolyzable tannins. Now this aren't susceptible to hydrolysis. The reason being there are no ester linkages, but most of them have what are called as carbon-carbon linkages. In this there are two categories: the condensed type, which have a flavan nucleus, and fluorotannins, which have a fluoroglucinol often linked to CC linkages or carbon, oxygen, carbon, what are called as your ether linkages. The last one, complex tannins, is a mixture of hydrolyzable and non-hydrolyzable tannins. So let's see the properties. If you see pseudotannins, they are easily water soluble because they are small. They do not tan leather, but because they have a good small molecular weight, they can bind to your tongue, salivary proteins and produce a very good astringent effect. On the other side, true tannins, because of their large molecular weight, their solubility goes on decreasing as the molecular weight increases. They have good ability to tan leather, and as they are high molecular weight, their binding ability to protein decreases, and so their astringency. So giving you a few examples of pseudotannin, the first one is your chlorogenic acid, other ones being your gallic acid, ellagic acid and so on. Whereas the same monomers may polymerize. So as you can see in this case, there is a central sugar moiety to which your gallic acid residues are there. So I'll just show you your gallic acid. You can see here there is one monomer of gallic acid. esterifying with another monomer of gallic acid, which turn esterifies with your sugars. Now this may polymerize in nature. I just remove this H and I just add one more gallic acid residue to it, and you can see the reaction can go endlessly. So this is one gallic acid which I added. Again I can remove one of the hydrogens and cause further esterification with an additional gallic acid group. So in same way they polymerize in nature to give you what are called as the polymers of gallic or ellagic acid. The hydrolyzable tannins can be easily distinguished by presence of a central sugar, generally which is a glucose moiety. The hydroxyl group of this sugar are complexed with monomers such as your gallic or ellagic acid, and in some cases hexahydroxy diphenic acid. They are highly susceptible to hydrolysis because of the ester linkage. So they are susceptible to acids, bases and the enzyme tannase. Now an interesting application of the enzyme tannase is, if you see these tannins, as the molecular weight increases, you will see their solubility decreases and then they tend to form colloidal mixtures. So imagine your tea, imagine your coffee, or imagine your red wine, beer. All of these examples are of extracts or water extracts containing tannins. So when you boil the tea, the tannins dissolve, but as the tea cools down, like if you want to sell cold tea or cold coffee, this tannins decrease their solubility with temperature and as a result form colloidal suspensions. Now as a marketeer, or if I want to sell this tea, my tea is no longer transparent. So if I have to render it transparent, some of this food industries, what they do is they add tannase to this colloidal suspensions. The tannase will cleave all the polymers, convert it into small molecular weight compounds, and the clarity of your tea, coffee, wine, beer can be improved. So this is an interesting application of tannase. Now if I have to distill it or dry distill it, on dry distillation all of these compounds give rise to what are called as pyrogallol. So pyrogallol is when you just cut off from this ester, you get a trihydroxy derivative which is called as pyrogallol. Now hydrolyzable tannins generally give pyrogallol on dry distillation. Two examples or two classes of hydrolyzable tannins are ellagitannins and gallotannins. In ellagittannin, the building block is hexahydroxy diphenic acid, whereas in gallotannins the building block is gallic acid. So on hydrolysis they will give you the same monomers. Ellagitannins, you will see actually gallic acid molecules are there, but they are joined by CC linkages. That is difficult to cut, but once they form the ester linkages are easily cleaved. So galloyl groups are present, but they are joined by CC linkages and hence cannot be broken. Whereas if you see gallotannins, there are gallic acid moieties or monomers which are joined to each other by ester linkages, so they eventually break and give you gallic acid. Few examples of ellagitannins are harda, which is a common fruit found in your Ayurvedic preparation triphala, which contains chebulagic acid, and another example is your pomegranate peel, which contains a ellagittannin called as punicalagin. Here if you see gallotannins example, they are found in nutgalls, they are found in clove, rhubarb. Below is a structure of tannic acid which is commonly found in your nutgalls and which is used for tanning purpose. Now coming to condensed tannins have a monomer which is a flavin type, and you can see the simplest monomer is a catechin. Now this monomers may form adducts. So imagine three molecules, they are joined to each other and they form polymers, and imagine this going on for about 2 to 50 such residues. They form a complex which are called as proanthocyanidins or condensed tannins. Now because the linkages here are CC linkages, they are not susceptible to hydrolysis, and often if you see the molecular weight, when they go to up to a 50 monomers joined together, they form a huge molecular weight complexes. In that case their solubility is very poor. So few classes or subclasses of condensed tannins are A type or B type. So if you see A type, they are polymers, but in addition to that you will see some linkages which are about C2 to C7. Okay. So this is the carbon number 1, which is O, and this is the carbon number 2. So from 2 it has formed linkage with C7. Whereas if you see B type, in B type the linkages are between C4, this is carbon number 4, to your C8. So your condensed tannins can be classified as A type or B type depending upon the linkages. The condensed flavin type nucleases can be again subclassified into many categories. The simple one is catechin that we just saw. If you see catechin and you can focus on this 3-hydroxy group of catechin, it lies ahead of the plane. Whereas if you see epicatechin, the next monomer which is involved in formation of condensed tannin, the hydroxy group is going behind the plane. So that's a little optical difference between catechin and epicatechin. Now if you increase one hydroxy group out here, you get what is called as catechin gallate. Same lies for your epicatechin, increase in hydroxy out here, you get what is called as epicatechin gallate. In some cases, this 3-hydroxy is involved in esterification reaction with a gallic acid moiety, and in that case it is called as epigallocatechin gallate. Now this is found in your tea and is investigated hugely for its anti-cancer effects. On dry distillation, they will give you catechol moieties. The last category of this are your fluorotannins. Fluorotannins are actually polymers of fluoroglucinol. So you can see here, fluoroglucinol is a dihydroxyphenol derivative. Now this monomers may form adducts via either ether linkages or they might form adducts via CC linkages. So they undergo polymerization. This class of compounds is not much found in plants, but they are generally found in algae, especially your sea kelps or some brown and red algae. A good example of those are ecol and fucofluorethol. The polymers of this range somewhere between 126 Dalton to 650 kilodalton in their weight. So thank you everyone for your interest, and here are a few references if you want to have an enhanced reading on this topic. Thank you.